Thank you very much for this introduction. I'm very happy to be there, and thank you to the organizers for the invitation. And it's a great opportunity for us to provide you a few insights of Gensight Biologics work. So Gensight Biologics is a public company, and this is the disclaimer. Gensight Biologics is a small biotech company developing gene therapy in inherited retinal diseases leading to blindness. And we have two main programs corresponding to two technology platforms. So the first one is the development of a gene therapy in a rare disease called Leber hereditary optic neuropathy. This is a devastating disease affecting young people in their 20s, 30s. And we have a technology which is called mitochondrial targeting sequence technology to overcome mitochondrial mutations leading to mitochondrial diseases. And we'll get into that. The second development program is about optogenetics. We are developing an optogenetic treatment combining a gene therapy and a stimulating device. And we are at early phase development in late stage retinitis pigmentosa with this combined therapy. Gensight was founded in 2012, so 10 years ago. And we have our headquarters in France, in Paris. And we have as well an office in New York. So this is an overview of a pipeline of a product portfolio in ophthalmic gene therapy. As I told you, our main program is a development of a gene therapy. The gene therapy is called Lumievoc in Leber hereditary optic neuropathy. So LHON is a mitochondrial disease, and there are several mitochondrial mutations leading to LHON. And the most frequent one is the mutation on the ND4 mitochondrial gene. It represents approximately 75% of LHON patients. And it is also the mutation leading to the most severe clinical form of LHON. And Lumievoc gene therapy is specifically targeting this mutation. So it's a devastating disease. It affects young people in their 20s, 30s, usually males. And these patients, it's a very acute disease, and these patients will become blind within 1 year. So there is a huge unmet medical need for LHON. We also are exploring the possibility to develop a gene therapy for other mitochondrial mutations leading to LHON, and especially we have been working on developing a gene therapy for another mitochondrial mutation on the ND1 mitochondrial gene for LHON. And we have another target which is not yet disclosed on a mitochondrial disease, with the same technology, the mitochondrial targeting sequence technology. For Optogenetic, we are developing in late-stage RP patients, so retinitis pigmentosa patients. The beauty of this technology with Optogenetic is that it's independent to the causal mutation. So as you know, retinitis pigmentosa is related to many genes and to many mutations. With this technology, we are aiming to be able to treat all late-stage RP patients. So late stage, meaning that they don't have any more photoreceptors in their retina, and we'll get into that. And we are also planning to expand this technology to more common disease, which is dry age macular degeneration or geographic atrophy. So let's focus right now on our lead development in ND4-LHON patients. So we are at late-stage development. 3 phase 3 pivotal studies have been completed, and we are in the middle of a process for regulatory approval in Europe, and we are aiming to get approval in Europe at the end of 2023. In parallel, we are preparing for the commercial launch of the product in Europe. So what is this technology, mitochondrial targeting sequences? And it's aiming to overcome mitochondrial mutations leading to mitochondrial diseases. Right now we are developing in LHON, but in the future we could develop for other mitochondrial diseases. So the Lumievoc gene therapy is an adenovirus type 2 containing the wild-type healthy ND4 mitochondrial DNA. It's injected via intravitreal injection because it's the best way to expose the targeted cells, which are the retinal ganglion cells which are affected in LHON, and the retinal ganglion cells are at the inner part of the retina. So when you inject in the vitreous, you really expose these cells to the gene therapy. It's a very easy way of administration, intravitreal injection. It's very well known by the ophthalmologists. It's not surgery, so it's a huge advantage. So once injected in the eye, the ND4 mitochondrial DNA is transcribed into mRNA in the nucleus of the retinal ganglion cells. And then these mitochondrial targeting sequences, you can see there MTS1 and MTS2, actively shuttle the mRNA to the surface of the mitochondria, where a healthy ND4 protein is synthesized and then translocated inside the mitochondria to restore energy production. So this is a unique technique to be applied to LHON in the future to other mitochondrial diseases. What are the key milestones? So for Europe, we are planning to submit our responses to EMA at the end of this year, so in October. This is actually the day 120 of the EMA procedure for registration. Then we'll get the feedback from EMA at the beginning of 2023, and we are aiming to get approval at the end of 2023. And for the US, we are right now discussing the best way to move forward in terms of regulatory pathway. We are a bit behind. So we have been working with centers of excellence in the US, in Europe, in Taiwan, and thanks to the patients and all the teams participating in this program. And you have here the associated publications related to this development. So now let's move on to another development, which is about optogenetics. As I told you, we are right now developing in late-stage retinitis pigmentosa. So these patients actually don't have any more photoreceptors, and you need to find another way to treat these patients because you cannot use the cells which are gone. So the principle of optogenetic is actually in these patients, in these late-stage RP patients, what you see on this picture is that the photoreceptors in gray, they are gone. So you cannot, work on it to restore vision. The principle is to use healthy cells, and in our technology we are using the retinal ganglion cells in blue on this picture. And the retinal ganglion cells are relatively healthy in late-stage RP patients. And our aim is to make the retinal ganglion cells act as photoreceptors, so in a way replace the function of photoreceptors which are gone because of the disease. To do so, we are using a combined therapy. So firstly, a gene therapy, which is injected via intravitreal injection, because it's the same principle. It's the best way to expose the retinal ganglion cells, the healthy cells, to the gene therapy. And the gene therapy encodes for a light-sensitive protein, which is then expressed in the retinal ganglion cells. And then we are using a stimulating device, stimulating goggles, which actually transform the external light stimuli into a signal that is recognized by the retinal ganglion cells, that can activate the retinal ganglion cells, and then the retinal ganglion cells are able to catch the visual information and to transmit the visual information to the brain to restore vision. So it seems to be a concept, it seems to be very basic research, but actually we could show that it works in patients. So we are right now running a phase 1/2 study called Pioneer in late-stage retinitis pigmentosa. It's a classical dose escalation study with 3 dose levels. We completed the 3 cohorts. We have an independent committee, a DSMB, reviewing safety data, and they advised us to use the highest dose in the extension cohort because the product is very well tolerated. These patients are unilaterally injected with a gene therapy in their worst affected eye, and we are working with 3 centers, 1 center in the US, in Pittsburgh, another center in the UK at Moorfield Hospital, and the 3rd one in France at Gensight Hospital. We recently released encouraging efficacy results from 2 patients from this trial. These 2 patients, actually, when they got into the trial, before getting any treatment, they had barely light perception. And with training with the goggles, with the gene therapy, with this combined therapy, they are now able to count and locate objects on the table. So for them, it's a huge improvement for their daily life. And we released the results of the first patient showing a partial recovery of vision. It was in May 2021, and it was published in Nature Medicine. And we released the results of the second patient. It was at the American Academy of Ophthalmology in November 2021. So, of course, these are few patients, but we are gathering data from other patients from the 3 cohorts. And here are the milestones for this program. So firstly, we got fast-track designation from the FDA in last October, showing that it is an innovative treatment that is aiming to cover a real unmet medical need for these RP patients. Our plan is to release the results of the 3 cohorts at the end of 2022. And then, while we'll be recruiting for the extension cohort, to provide the full results of the trial at the end of 2023. And the next step will be to move forward to a pivotal study in late RP patients, and we are planning this trial at the beginning of 2024. In parallel, we would like to expand this technology to a more common disease, which is dry age macular degeneration or geographic atrophy, and we are planning to launch a phase 1/2 study in this population at the beginning of 2024 as well. Thank you for your attention.